Hi, my name is Lerfos Jainus, and in this video, you will learn about population genetics. What is population genetics? Population genetics is the study of how populations change genetically over time. It also related to study of the change of frequencies of alleles within a population. It emphasizes on the genetic variation within a population. So, how to calculate the allele frequencies? The allele frequencies can be calculated or can be determined by using gene pool concept and Hardy Weinberg equilibrium. First, let's discuss about gene pool. Gene pool consists of all copies of every type of alleles at every locus in all members of the population. Diploid species or diploid organisms, for example, consist of 2 alleles for every gene. For example, suppose a population has 320 plants with red flowers, 160 with pink flowers, and 20 with white flowers. For this particular population, the total number of individuals within this population is 500. So, the total number of genes that will control the flower color within that population is 1000 copies of gene. Now, how to calculate the allele frequencies? The allele frequencies is the ratio of the total number of given allele. For example, to calculate the allele A, we use the equation total number of dominant allele A divided by total number of all alleles. And to calculate the allele frequency for recessive allele, we use the equation total number of allele A recessive divided by total number of all alleles. For example, a study was done on 10,500 rats, focusing on the fur color determined by dominant allele C and recessive allele C. The number of individuals with the genotype homozygous dominant C is 6,300. The number of individuals with heterozygous genotype CC is 2,200. And the individual with genotype homozygous recessive C is 2,000. We can use gene pool method or gene pool concept to calculate the allele and genotype frequencies of the population. The allele frequency for C allele dominant is equal to total number of allele C divided by total number of all alleles. From the question, the allele frequency for C dominant allele equal to 2 times 6,300 plus 2,200 divided by 2 times 6,300 plus 2,200 and plus 2,000. That will give us 14,800 divided by 21,000 equal to 0.705. To calculate the allele frequency for C recessive, total number of alleles C recessive divided by total number of all alleles. From the question, 2 times 2,000 plus 2,200 divided by 2 times 6,300 plus 2,200 plus 2,000. That will give us 6,200 divided by 21,000 equal to 0.295. The total allele frequencies for dominant allele and recessive allele equal to P plus Q equal to 1. The next method that we can use to calculate the allele frequencies is we can use the Hardy Weinberg law. According to the Hardy Weinberg law, the frequency of alleles and genotypes in a population will remain constant from generation to generation. provided certain conditions are met, or in other words, the population is in genetic equilibrium. There will be 5 assumptions or conditions for Hardy Weinberg. Number 1, large population size. Number 2, random fertilization or random mating. Number 3, no mutation. Number 4, no migration. Number 5, no natural selection. Now, we will discuss the 5 assumptions of Hardy Weinberg law for genetic equilibrium. Number 1, large population size. Very large population size so that genetic drift can be avoided. Genetic drift is the chance of fluctuation in the gene that can cause phenotype frequencies to change over time. If the size of the population is very large, the small changes or the chance of fluctuation in that gene can be ignored. The genetic drift is not significant in a very large population. The second assumption is random fertilization or random matings. That's involved the mating of individuals regardless of genotype and phenotype. All of the individuals within that population will have an equal chance of being selected and pass the genetic into the next generation. In other words, all individuals within that population will have an equal chance to reproduce and being represented into the next generation. Because if individuals pick mates with certain genotype, the random mixing of gametes required for Hardy Weinberg equilibrium does not occur. Assumption number 3 is no mutation. No mutations because by changing one allele into another, mutation will alter the gene pool. This will change the allele frequencies. Assumption number 4 is no migration. No migration that causes the gene flow due to immigration into the population or emigration out from the population. Death also is a type of emigration because death will remove the gene or allele permanently from the population. Assumption number 5 is no natural selection. It means that all genotypes are equally fertile so that no natural selection is taking place. It also means that all individuals will have an equal chance to survive and reproduce so that it can pass the genetic material from generation to generation. We can calculate the allele and genotype frequencies by using Hardy Weinberg equation. To calculate allele frequencies, the equation is P plus Q equal to 1. P is the frequency of dominant allele, Q is the frequency of recessive allele, and 1 is the total of allele frequencies within that population. If the population is in genetic equilibrium, P plus Q is always equal to 1. To calculate the genotype frequencies, the equation is P squared plus 2pq plus q squared equal to 1. P squared is the frequency of homozygous dominant individual. 2pq is the frequency of heterozygous dominant individual. And q squared is the frequency of homozygous recessive individual. 1 is the total of genotype frequencies within that population. The following are the helpful tips that we can use to calculate alleles and genotype frequencies using Hardy Weinberg equation. First, from the question or from the situation, find the number of recessive homozygous individual first. This is because the recessive phenotype consists of homozygous recessive Q squared, while dominant phenotypes, it may be, consist of homozygous dominant P squared or heterozygous dominant 2pq. Tips number 2, find or count or determine the population size. It may be in the form of number, percentage, or ratio. Tips number 3, divide homozygous recessive trait by the population size to get the Q square. Tips number 4. Square root of Q square to get Q. Tips number 5. From the equation P plus Q equal to 1, we can subtract Q from 1 to get P. P equal to 1 minus Q. Tips number 6. Since P and Q are already known, we can use this information to solve for 2pq and p square. We will have the equation p square plus 2pq plus q square equal to 1. Tips number 7. Always include the equation p plus q equal to 1 and p square plus 2pq plus q square equal to 1 in your calculation. And here is some of the example that we can use to calculate the allele frequencies and genotype frequencies. Example 1. In a population of 500 wildflowers, 20 are white flowers are having recessive phenotype and the rest have dominant phenotype. If the alleles involved are dominant A and recessive A, and there are 320 homozygous dominant plants and 160 heterozygous plants. What are the frequencies of heterozygous genotype individual, dominant and recessive phenotype individuals? For this type of question, we can use gene pool method and Hardy Weinberg equation to solve this question. Let's try the gene pool method. From the question, the total number of allele is 2 times 20 plus 160 plus 320 equal to 1000 alleles. To calculate the frequency of allele A recessive, total number of allele A recessive equal to 2 times 20 for homozygous plus 160 for heterozygous. That will give us 200 alleles A recessive. So, the frequency of allele A recessive is 200 divided by 1000. That will give us 0.2. So, the frequency of allele A recessive is 0.2. To calculate the frequency of dominant allele A, total number of allele A 2 times 320 for homozygous plus 160 for heterozygous. That will give us 800 alleles A dominant. So, the frequency of allele A equal to total number of allele A dominant divided by total number of all alleles equal to 800 divided by 1000 equal to 0.8. So, the frequency of allele A dominant is 0.8. Next, to calculate the frequency of AA genotype, which is heterozygous, total number of heterozygous divided by total number of population, 160 divided by 500. That will give us 0.32. To calculate the frequency of dominant phenotype, total number of dominant phenotype divided by total number of population, which consists of homozygous dominant and heterozygous dominant. 320 plus 160 divided by 500. That will give us 480 divided by 500 equal to 0.96. To calculate the frequency of recessive phenotype, total number of recessive phenotype divided by total number of population. The recessive phenotype consists of homozygous recessive AA. 20 divided by 500 equal to 0.04. Now, we will calculate the allele frequencies and genotype frequencies using Hardy Weinberg equation. P plus Q equal to 1. The frequency of homozygous recessive individuals is Q square. Q square equal to 20 divided by 500. Q square equal to 0.04. q equal to square root of 0.04 equal to 0.2. Since p plus q equal to 1, so p equal to 1 minus q. P equal to 1 minus 0.2. P equal to 0.8. So the frequency of heterozygous individuals is 2pq equal to 2 times 0.8 times 0.2 equal to 0.32. To calculate the frequency of genotype and phenotype, we can use the equation p squared plus 2pq plus q squared equal to 1. The frequency of dominant phenotype equal to p squared plus 2pq. 0.8 squared plus 2 times 0.8 times 0.2 equal to 0.64 plus 0.32 equal to 0.96. The frequency of recessive phenotype is q squared. Total number of homozygous recessive divided by total number of population. 20 divided by 500 equal to 0.04. If the population is in Hardy Weinberg equilibrium, p squared plus 2pq plus q squared equal to 1. So 0.64 plus 0.32 plus 0.04 equal to 1. So this population obey the Hardy Weinberg principle, and this population is in Hardy Weinberg equilibrium. So that's all for now. See you again in the next video. Thank you.